Hello, everyone. My name is Viduthalai Rasheedkhan Regina, and I'm a senior research fellow from Singapore Center for Environmental Life Science and Engineering in the Nanyang Technological University in Singapore. So in this talk today, or the lecture today, I'm going to introduce you to the human skin microbiome. I know a lot of folks here are from a zoology background, so I'm going to keep it from a very basic level so you can understand what's going on with our skin, right? So usually when we study our skin, it's basically from the standpoint of the human cells, right? So, but then lately in the last couple of decades, there is a vast understanding that it is not only the human cells makes the skin and its function basically. So the function of the skin heavily depends on the microbes on our skin. So this is a very latest understanding, and there is a lot of therapeutics and all that are actually being developed based on this knowledge. So I think this is going to be very useful for you guys to understand the microbes a little bit and how they are actually helping the human system, or not only the human system for that matter. It applies to any animal basically, right? So okay, so let's go into what we are going to look at it today. So we are going to have, so this is kind of the outline of my talk today. So here first I'll set something straight, because like a lot of, like many evolving fields, initially the terminologies are going to be a little bit redundant, right? So microbiome field is also a very new one, relatively new, and the terminologies are going to be a little bit redundant also. So I want to just give you a sense of what are the terminologies that are going on in the field, and then I'll introduce the microbiome in and on our skin and how diverse they are, what is the variability between people, etc., and how the microbes in our skin interact with each other, as well as they're interacting with our human cells. And then what are the possible, based on this knowledge, what are the possible interventions that we can actually do, and how things are progressing at the current environment, right? Okay, so coming to terminology, this is a general terminology everyone know: microflora, microbiota, and microbiome. So microbiota and microbiome especially is a little bit of a latest in these three. Microflora is, we know that it's a botanical term, and microbes were basically looked at that from a botanical point of view also, especially bacteria and all that, traditionally speaking. So microflora is basically micro, which is like micron sized or below, and flora, and biota is basically the organisms. When you're actually working with organisms, then it becomes microbiota. And microbiome, you can actually take it into two different ways. Microbiome as in ohm, if you think about the omics, right? So proteomics, transcriptomics, genomics, and all that ohm. So if you work with these kind of data set, then you can also call it as microbiome. And also the microbiome as in a combination of a lot of microbes in one space in time, that is also called a microbiome. So some people, when you look at the literature, they do use it redundantly. Some people kind of use mainly microbiota and microbiome, the difference between them. So it's a little bit of a gray area there. But just for our understanding, microbiota means like you are talking about the individual microbes that are actually within a system, and microbiome represents the whole whole system per se, right? Okay, so I'm just explaining here. So these are basically organisms, and when it comes to omics, then it is a metagenome, like the whole thing, right? And then microbiome generally means it is a community that we are talking about. So this is more and more maturing, and latest publications, people do write microbiome in the sense of a community, right? So this is how it is. So just I want you guys to have a think about. Sorry, it's a little bit of a too quick to disclose, but then Yeah, I just wanted to, have you think about, what are the domains that are, that the microbes constitute, right? So is it only bacteria? Is it archaea and eukarya? So these are the three major domains of life. And where do you think the microbes are actually placed, right? So basically, if you think about it, microbes actually spans all the three domains, right? So there are also eukaryotic microbes, and archaea are of course microbes, and archaea is where is our uh, the common ancestor for most of the eukaryotic organisms are from archaea, and then bacteria is a separate clade in the domain, right? But all these are dominated by microbes, actually, in fact dominated by microbes. So there is a lot of microbes out there than any number of animals in the world, right? Okay, microbiome in and on our skin. So what is actually the microbiome, or how does it look like, or how is it actually spread across the human body? So this was a very good study that was done in 2015, where they actually sampled the human body from all the regions, almost every region of the human body, and then they basically sequenced the whole genome, and then understood that, and then basically made a list of microbes that are present in these body sites. And what you're seeing here is a Shannon index, which is a diversity index. And the red means that it is really highly diverse, and then blue means it's low diversity. And as you can see that most places of the body is very, very complex and dynamic microbial community that is out there, right? So you can see that almost everywhere you can see microbes, right? So this is on top of the skin. So is it just present on top of the skin? And then there was another study that I wanted to show you where they took a biopsy. So, and then you can basically, this is the depth of the skin, and you can see that there is a part of the hair follicle that is present. So they took a biopsy, and this is in millimeter, right? So up to 4 millimeters. And this is from a face, and this is from palm, right? So, and then what they did was they took slices of individual areas in the biopsy, and then they basically used a marker gene, which is 16S rRNA. These are specific for a bacteria, and then they basically used a qPCR, quantitative polymerase chain reaction, to understand like how much bacteria are present in these different layers of the cell. And as you can see that, the bacteria extends to up to like 4 millimeters of our skin. So they are just not passively just being on the skin just because they are coming from the environment or something like that. So they are basically part of our skin in a very deep layer. So one more line of evidence is this study where they actually took again a punch biopsy of a hair follicle, and then they used immunofluorescent dyes to basically target human cells and bacterial cells. And the bacterial cells were targeted by this green fluorescent dye. And then you can basically see that from all these different regions in the depth of the skin, you can see that the bacteria colonization is present. So what does it mean is that they are not just present on the surface, but they are integral part of our skin, right? So this is what I wanted to convey. And that our skin basically connects us to the environment, right? So skin is actually protecting us from the environment, not only protecting because it has a lot of sensory neurons and so on, so it can sense a lot of things from the environment. I mean, majorly if you say that temperature and the pressure and pain, etc., it can sense, right? So it is basically the connection between the environment and us. And basically the microbes on our skin are also the ones that are connecting us to the environmental microbes, or the microbes in the environment and us, right? So if you take a look at this plot where the individual dots, right, there are colored dots here, right? So individual dots are from different samples from the environment, right? So each different sample represent a mix of microbes, a community of microbes. There may be millions of microbes in here, right? So they were all put together based on how similar the community is between different different samples, right? So when they are very close together, let's say this is a red one, right? So red is from a fly, and then blue is from a or light red or pink from the soil. And when they are actually together, that means that the most microbes in the community are very similar to most microbes in the soil. So they basically overlap. That's why they are together. So this is from the environment. And the other side, they took it from animal systems, right? From rats and human gut, and then also skin. What we were able to see was that if you see these animal gut microbiome that is basically in the system, they're actually very much separated from the ones that are in the environment. But then the skin comes in between them. So basically, as I said, our skin basically connects the human body to the environment. The microbes in our skin is actually connecting the microbes from the environment as well as from our gut. So it's kind of a continuum. So this is a very meta study. It's a very big study that was done from from a lot of different samples around the world, and you can see that plants, humans, termites, and everything. And then you can see that yellow one, the yellow patch, it is not very clear here, but then there is this yellow patch that is in the middle of everything. So basically this is connecting every single entity on the planet, right? So they are really important in that way, that they are connecting us to the environment. So basically we know that the skin is functioning as a lot of different things. As I said, like, it can sense the temperature, pressure, acidity, all that things are going on, right? It is very robust. It is also giving protection from the pathogens, etc. But it also has a very functional immune system, right? This immune system is completely different from actual body's immune system. They do have connections, but then they have their own immune cells, their own set of cells that are present in the cells. That's because they are actually exposed to the environment, and the environment has a lot of different active microbes. And as you can see that these microbes are actually educating our immune system, and the immune system and these microbes has a connection, and that is actually how the whole system is working. So basically the human immune function is not complete without the microbes on the skin, because we know that these microbes secrete specific molecules that actually triggers our immune system, that then produces AMPs, like antimicrobial peptides that will kill the pathogens. So basically the microbes on our skin is educating our immune system to to kill pathogenic microbes and to keep good microbes, commensal microbes within us, right? So next thing I want to tell you is, like, okay, the skin is really composed. I mean, a lot of microbes are present on our skin, and how diverse they are, like how diverse and they are, and how big of the difference between individuals, right? When it comes to diversity, I mean, it's a heavily diverse population of population of microbes, but then it is not very diverse when it comes to oral cavity or our gut, right? So what does it mean is that you can see that there are three kingdoms that are present: eukarya, bacteria, and viruses. And then within the eukarya, the majority of them are fungus, and within the fungus, Malassezia is the majority of the species that is present in different regions of the skin. And then when it comes to bacteria, so these three bacteria, Propionibacterium, Corynebacterium, and Staphylococcus, these are the major ones that are colonizing the skin, even though there are other bacteria that are present. So you can say that these constitute the core skin microbiome, but then there are other microbes that are also diverse, right? So the diversity is a little bit high here. Now coming to the variation, right? So diversity is actually what are the different types of bacteria that are present among the whole community of humans, right? Variation is basically how different my microbe is from my partner's microbes or is from your microbe. So that's the variation, right? So I just want to highlight one study where they basically sampled the keyboards of different people's computer, right? And then they came up with this. So they accessed the keyboard, and they also used the fingertip. They sampled the fingertips of their people, and then they analyzed these results. And you can see that all these individuals, the dark ones, the filled ones, are actually from the keyboard, and the open ones are from the fingertips, right? So you can say that see that they are all clubbing together. This is not surprising, because you are typing in the keyboard. Of course, the microbes on your skin, on your fingertip, will be there. The surprising thing here is that in these three individuals, they are separated as different entities, right? So they are clustered together in different space within this multivariate plot. So each individual dot represents like hundreds of microbes, right? And this hundreds of microbes is not very similar to this hundreds of microbes, and the same here, right? So this is what variation means. Variation is like it is very specific to people, and people have used this to even forensics. Like, if they sample a place, and if they know that, if there is a particular community, they can even track it back to the person who was there, right? So this can be tracked. And then also, like, there was a very fun study where, they sampled the belly button of individuals who came to a conference, and then they were able to track back the community of microbes in the belly button to wherever they have gone for vacation or whatever they are, because everywhere you go, you shower, and the belly button has the capacity, because it's just a small pit. It can accumulate organisms. By sampling that, they were able to say that, okay, they have gone to Hawaii in this place because Hawaii has this signature microbiota that is there, and that was present in the belly button. So it is very specific for individuals, as well as you can track back to specific areas or places and things like that, right? So that's where the variation comes in. And the most, so I told you, right, the stool sample is basically a representative of the gut microbiome, right? So when they sample the mouth, stool, and the skin, the most variability comes from the skin, right? So the most diverse population is from mouth and stool, right? So they have multiple number of bacteria, but then if you take you and me, most of our microbes are similar between you and me, but if you take our skin, they are very different, right? So because we are getting exposed to different environments and so on, so that's what it meant. So this plot is basically, every single turn means that this is a data point. So this data point actually is different from this data point, and so on. So they made a continuum suggesting that, these are very compact compared to the skin. That means the skin is most variable in community, and that's also very important, because when it is very much variable, it is able to educate the immune system very, in a good way, right? So yeah, again, this is all variability in the sense that, it is variable in the, it's not in the species level, it is also variable in the strain level. So if you take these two different microbes, Propionibacterium acnes and Staphylococcus epidermidis, individual color means that there are different strains of these microbes, and these are 4 different individual, and you can see that how different their microbes are at the strain level, right? So it is a very, very variable community. So what we saw is that even though the diversity is a little bit less than mouth and stool, the variation is quite high, right? And also, not only it is variable, it is also very, very stable, right? So what they did was they took a sample from several individuals from different regions of the skin, and then they tracked actually for like full year, right? So what happens, you have different seasons, you get exposed to different seasons, and then you do meet a lot of people, you shower every day, you put shampoo, soaps, and all that. But the baseline was that all through this time, the microbiome did not differ, as in every individual was able to protect or keep the microbiome, or the microbiome is stable for every individual, and the differences between the individuals remained the same, right? So it basically again reinforces the thing that, we have our own signatures, and this can even be tracked as a forensic thing, right? Okay, so now we know that it is diverse, and then it is very variable, and then it is very stable over time, despite all these interactions, all these exposures that we are getting, right? So now going into the key members, as I showed you, like there are like these 3 organisms that are very important or key members of this microbial community. Even though you have a lot of different organisms, these 3 stands out as the most dominant species in our system, which is Propionibacterium acnes. The new name is actually Cutibacterium, but it's an old name, but I'm just keeping the convention going. And then you have the Staphylococcus epidermidis, and then Malassezia restricta. So these are the 3 organisms. The interesting bit is that these 3 organisms are really good bacteria for us. At the same time, these bacteria can also be a clinical, can also cause infections and clinical outcomes. Like Propionibacterium is associated with clinical acne, very strong acne as well as implant infections. Staphylococcus epidermidis is also associated with a lot of implant infections where implant failures can occur. Malassezia is associated with like dandruff, seborrheic dermatitis, folliculitis, etc. So it is a very interesting thing that, these are the bacteria that are really good for us. At the same time, if something happens bad, they actually can act as a pathogen. So there is a field called emerging pathogen where a normal bacteria or a commensal bacteria or good bacteria can emerge as a pathogen. So I also have another talk in this series if you are interested to take a look at it, right? So these three bacteria, we know that they are important. So how do they are they interacting between each other? Right, so this is a question that we are also looking into, like I'm also interested in when it comes to research. So what we did was basically we grew the bacteria separately as well as we grew the bacteria together, right? So what we saw in different conditions is that when they are together, when the bacteria are together, they are able to form a really strong and a resilient biofilm or a community when compared to when they are alone actually. So you can see that SAP is alone, C. acnes is not able to form a very good growth here, and then when they are together, it is actually producing a lot of growth. So that means like they are basically interacting between each other. And what we also saw that is they are very resilient when they are together. For example, this is C. acnes alone. C. acnes is actually Cutibacterium acnes, and S. epidermidis alone. And what how do you read that is like when we grew these bacteria and then treated them with antibiotic, 60% or 50% of the bacteria were dead or went away, right? And then with the S. epidermidis, it is actually not very, it is not actually susceptible to the antibiotic treatment. So when we put them together, the whole community was protected because this one is able to protect this. So they have a interaction going on and the whole co-culture was protected. And when we imaged it, what we saw was that the blue ones are the P. acnes and the S is the S. epidermidis. So they are actually together. So what initially we thought was all the C. acnes, the blue ones, would have been died, and S. epidermidis would be the only one that is surviving here. But what we saw was both of them were coexisting together. So which means the protection that it is not protecting itself, but it is actually protecting the co-member in the community as well, right? So that is how they are. When things are together, they are very resilient to environmental stress and so on. So diversity is the key here. So if you have a very diverse and good community, you are protected from a lot of different ailments, etc. Next thing is like what we know about the skin microbiome and how it affects our health, right? So so as I said, like before I told you, right, when even when the two bacteria are together, they are more resilient, right? And when the diversity is lost, then it is actually a problem. So for example, we had a very good environment out there, you have a greenery and all that, so you are exposed to a natural system, and then now over time, because we are more and more people are moving into cities, more and more cities are expanding, we also lose this diversity of not only the trees and plants and trees, it also leads us in the direction of losing our own microbial diversity, right? So the loss in diversity actually has shown some increment in allergic and autoimmune diseases, right? One of the studies I want to highlight is about it was done on dogs in Finland. So what they did was they took three populations of dogs, and these dogs were like one population was born in rural area and then raised in rural area, and then one cohort was like born in urban and then raised in rural, and then the other one is born in rural and raised in urban, right? So sorry, four actually. So these four combinations, one is the extreme where rural born, rural raised, urban born, urban raised, and then urban born, rural raised, rural born, urban raised. Okay, so when they looked at the community was quite different from each other, all these four different communities. So for example, this one is urban born, urban raised, the black one, it is different from rural born, rural raised. And then rural born, urban raised is different from all these two, and then urban born, rural raised is also different from these ones. So we do see that there are three different community of microbes. So then what they did was they actually correlated this with the incidence of allergies in these dogs. And what they found was that when the dogs was born in the rural area and then raised in rural area, they have less prevalence of allergic, sorry, I have to show you this one, the blue one. So when they are born and raised in rural area in both places, though, so they have less prevalence of allergic symptoms. And when they are born in the urban area and raised in urban area, they have high prevalence of allergic symptoms. And then you can, as you can see, rural born dog, when it went to urban areas, the prevalence is a little more than rural born and rural raised, but less than urban born and urban raised. And as you can see, urban born rural raised has less allergy than urban born urban raised. So what does it mean is actually the environmental exposure also matters a lot when it comes to allergies and diseases, because when you have more microbes in the environment, your skin has the opportunity to interact with more microbes, and that's how it actually educates its immune system. And when you have a good immune system, you don't have allergic symptoms, right? So this is what it emphasizes. So it is very important to have a very good diverse microbiota on our skin for our immune system to work better, right? So what happens actually here, right? So for example, the urban dogs and so on. So, and especially when it comes to allergies and all these different conditions. So we have a healthy microbiome or microbiota where you have all these different types of microbes that are present. What happens is generally for a healthy individual, there is no problem. But then when there are triggers, like, when there are pathogens coming in or pollutants or whatever, then the microbial composition completely differs from the healthy one, right? That's called a dysbiotic state. That is a disease kind of state, right? And we have a lot of evidence on this. Like, this is a healthy skin where you have different types of bacteria here, right? They have a label here, and then there is a condition called atopic dermatitis, right? So people have eczema and they have itches here. Most children have these days, urban children have a lot of these conditions, and you can see that the staphylococcus is actually overpopulated in this community. So this is a dysbiosis, and the other one is acne. So healthy follicle has a lot of bacteria that is present, and the composition basically changes, and then the acne follicle has this irritated area and then bulging and so on. So we understand now that it is not the skin's problem, it is actually the microbes that are associated with these conditions, right? And these microbiomes are actually contagious also. So what does it mean is that so when there is a unhealthy microbiome, if there is transplanted or somehow moved to a healthy microbiome, then that can also affect basically. So this was a study that was done on mice where they transmitted infected community Leishmania infection, and then they moved it to a regular mice, and then what they saw was that the community differed, the microbial community differed, and then the pathogenicity was acquired by the other community. So usually when we talk about pathogenesis, we usually talk about single bacteria or single microbe infecting a system, right? But then what we know is the microbiome itself can be transplanted, and this unhealthy microbiome can actually move a healthy microbiome to unhealthy, and the other way around is also true, right? Okay, so this is again, the data from this particular experiment where it was shown that the native microbiome is different, and then the lesion, and then when they were moved to a healthy mice, the community actually went up like the lesion, right? So yeah, basically what I said, but this has more data into it. If you want to take a look, you can pause the video and take a look, right? So interventions, based on this knowledge, we also have developed a lot of interventions, right? Since it is contagious, as I said, it can also be used as a transplanting tool, right? So there is a very good experiment on the gut microbiome where they have showed that and in the gut microbiome, you have the obesity is also associated with the microbial dysbiosis in the gut. So what they did was basically they transplanted the microbes of a obese mouse into a lean mouse, right? and then they saw that the lean mouse was able to develop obesity because the microbes were different, right? Um, so um, uh, Give me a second. So I think that it is the other way around, right? So if you are transplanting a healthy microbial community of a healthy mice to an unhealthy one using fecal transplant, basically the obese mice became lean and the microbiota actually resembled the healthy one, right? So this is actually being used in some infections like C. difficile infections, where microbiome transplant through fecal microbial, it's called fecal microbial transplant, FMT. So basically the fecal material from a healthy person is transplanted to an individual who has this infection. So this has been done before, right? So and this is actually going on as a treatment in the US, for example. So can it be done on skin? Can we apply microbes on the skin to treat infections on the skin? So there has been a lot of research, and we are very close to seeing one product from this company called Dysbiosis, which is from it's a Belgian company, and they have just released their data from their clinical trials that a live skin probiotic, a live bacteria that is actually going to be supplied in a tube, and you can apply it for your skin for acne and other things that can improve the skin health, right? So it is possible, and there are more and more things that are going to come into play, right? So this is where I'm going to stop here, and I hope I have given a very good, a little bit of an introduction for the microbial role on the skin, and how it is important for the skin's immune function, and how diverse they are, and how variable it is between individuals. And I hope you can dig deeper. And if you want to learn more about this, this is my website, drvidu.wordpress.com. You can visit my website. If you want to contact me, you can contact me for more information, etc. Right? So I'll stop here, and if you have questions, you can contact me, and hope I was able to give you some introduction to the microbiome. Yeah, thank you.